Hello everyone. Today I will deal with the topic of plant growth and development. Growth is the most fundamental and conspicuous characteristic of a living being, and in the case of plant, how it will be, that's what we will be try to understand in today's class. When a plant lives in any given environment, it constantly puts effort to grow. Its growth begins as a single cell, that is zygote, which then forms tissues, organs to become a self-sustainable individual. And it is a highly ordered succession of events. During these events, there is a progressive increase in its structural and functional complexities. These events are self-programmed, that is genetically programmed they are, and tightly regulated and adaptable for environmental fluctuations. The events in plant's life cycle are distinct and prominent during specific stages. Through them, we try to visualize the growth, and it is a result of complex events. Let us study some of the prominent events which happens in the life cycle of the plant. So there will be a seed formation, seedling and sapling, and thereby give rise to mature plant which gives flowering and senescence. And each of these events are influenced by many factors. An overview of these manifestations, collectively called as growth in layman's terminology, or development in more profound way. And to understand growth and development along with the differentiation, we have to understand it as a development as an umbrella term which tells about the growth and differentiation as the sums of that particular aspect. And development is the most visibly manifested as changes in the form of an organ or organism, such as the transition from embryo to seedling, from a leaf primordium to fully expanded leaf, or from the production of vegetative organs to the production of floral structures. Now we will move on to study growth and its nature. Growth is a quantitative term related only to changes in size and mass. This may be observed as increase in length as in young plants, or width by girth in trees. For cells, growth is simply an irreversible increase in volume. For tissues and organs, growth is normally reflects an increase in number and sizes of the cells. So growth can be accessed by a variety of quantitative measures, and dry weight and fresh weight are the more frequently used measures. Here, dry weight is nothing but weight of plant without water, so usually it comes very less, and weight of the water with water is called as fresh weight. And choosing which weight to consider depends on its credibility of each method in individual cases. And since growth is quantitative in nature, it can be measured by choosing proper instruments which can measure size in its different manifestations. Some of the very important growth measuring devices are ruler, tape, oxynometer and crescograph. Let us understand another accompanying term, differentiation. Differentiation occurs when cells assume different anatomical characteristics and functions or form patterns. For example, if we consider embryogenesis, which begins from a zygote and passes through octate shape, heart shape, torpedo shape, and to become a mature embryo. In all these cases, it has undergone cell division, elongation, differentiation in pre-programmed manner. In all these events, not only size, that is growth, changed, but also appearance and functionality is also changed. All these changes we can trace qualitatively. And meristems play a very vital role in the sequence of events. they bring polarity as root meristems and shoot meristems, which establishes axial patterning in the plants. And followed events of development led by the differentiation with established radial patterning of the organs. The spatial specific positions of various structures will be established, like stem and root anatomy, where epidermis, cortex, pith, everything will be in a sequential manner. When we observe a tree, we can notice that in initial years it grows in length, that is in height, where the differentiation will be facilitate this particular polarity. Later it starts expanding in its girth. This type of proliferation is termed as vegetative growth, and it will be of indeterminate type. That is, no end for its growth till the senescence. Among these type of differentiation, meristem is the most basic one. They ensure one lineage of cell will retain their ability to differentiate throughout their life, like root apical meristems, shoot apical meristems, and cambiums. Development of the tissue is a result of division, enlargement, differentiation, and maturation of individual cells. It is obvious that the tissues like parenchyma, which is a simple permanent tissue, has lost its ability to divide. But plants shows the capacity of regaining their metasomatic activity, and it is called as de-differentiation, and which is seen in the form of an interfascicular cambium, which is grown in between the fascicular cambium and cork cambium, which grows in the cortex region of the during the secondary growth in the plants. De-differentiated tissue becomes capable to produce mature tissues, which is called as redifferentiation, as in case of secondary xylem, secondary phloem. All these properties are indicating plant cells are totipotent. While we observe the tree, we can also notice that while overall growth of the plant is conspicuous, there will be constant loss of leaves, seasonal appearance of birds and flowers. These organs will be a result of determinate differentiation, and it is derived from the vegetative or indeterminate meristems only. So the flow of the process is as follows. The vegetative meristem give rise to axillary buds give rise to leaf meristems, and auxiliary buds sometimes can also give rise to inflorescence meristems, and leaf meristem give rise to leaf, and inflorescence meristem give rise to inflorescence, and some or flower meristem, and flower meristem give rise to flower. So this is how the sequence will be always definite. So development of a plant is both qualitative and quantitative process, and this physiological process requires many inputs, and the relationship can be briefed in the following manner. So here we can see the development as the central idea in the plants, and growth and differentiation are some together they will form the development. And this development is not only intrinsic in nature, but there are extrinsic factors are also there. So there are two types of factors which will influence it, and internal factors and external factors. And while noticing the internal factors and external factors, we can notice that genetic factors and phytohormonal factors play a very important role in plants' growth and development, whereas nutrition, water and light, oxygen and temperature will act as external factors in the plant's growth. And these are all the various overviews about the development in the plant's physiology of growth. Hope my lecture was useful to you. Thank you for listening.